Okay, is this working? Okay, cool. So let's get started. Thank you everybody for coming. So it's an event. It's the, I forgot, 5th colloquium of the Center for Computational Life Sciences. It's a platform for all of us to come together to talk about interdisciplinary topics across computer science, biology, and engineering. So if you don't get emails and you haven't gotten this announcement and only heard it from your next door neighbor or office mate, sign up for the email list so you don't miss the activities. There are hackathons, there are like educational sessions that are being organized by Richard, for example. So please, please participate. I think this is a unique opportunity for us to interact. So otherwise, it's my great pleasure to introduce Gunnar Rätsch. Thank you for coming. Gunnar Rätsch is a professor at ETH Zurich. He studied physics and computer science. He received his PhD from the University of Potsdam. I got the speaker's note just in case. He did his postdoc in Australia and at the Max Planck Institute in Tübingen. This is also where he started his first research group working on genomics and method development and machine learning development in plant genomics and Arabidopsis. From there he moved on to the Memorial Sloan Kettering Cancer Center to work on machine learning methods for cancer patients, and eventually landed at ETH Zurich, I think about 10 years ago now, as a full professor in computer science. He's the head of the Institute of Machine Learning and is leading the biomedical informatics group. So this group is working in areas like time series analysis for ICU, compression, storage, searching of petabyte genomic data sets, as well as analysis of heterogeneous cancer data sets. So thank you so much for coming, and the floor is yours.
Good, I need this thing. Just a second. Okay, can you hear me okay? Yes. So it's a great pleasure to be here. Thanks so much for the invitation. Kyung and I, we have worked for a long time. So when I moved to MSKCC, I think Kyung was my first postdoc I hired there. And we did a lot of cool projects together. And one of the projects I'm going to talk about today is one of these, which we did at ETH. But let me first give you an overview of what else we do. Okay, so here's a little overview of what we do. We are quite diverse in the kind of methods and topics we work on. So, I mean, my mindset is I'm a computer scientist and I'm trying to find interesting methods which have a large impact, hopefully, okay? And we try to have usually a component which has a new methods part and a relevant application domain part. So it's not just pure methods and it's not just pure applications. So we try to go this line in between, right? Okay, so one topic is, that's what I came for GCB, the German Conference for Bioinformatics. I gave a talk there. And we work on very large scale data structures for genomics. And the idea there is that you have a big pile of genomics data, and there is the sequence read archive, for instance, and you would like to search in it, and you can't. It's impossible because the data is so large. It's essentially as large as the whole internet, data-wise. And we have developed algorithms which can index this, compress it extremely well, goes from 70 petabytes down to maybe 150 terabytes, and then you can actually search in it efficiently. So within a few minutes, you get a full text search result back of this kind of data. So I'm not going to talk about this today. Today I'm going to talk about cancer and multiomics. This is the topic which I chose. There's other things. Yeah, if you would like to try this, go to metagraph.ethz.ch. The other topic which I'm not going to talk about is our machine learning work for modeling temporal data. So we think about medical data, medical time series, multimodal heterogeneous time series data, and we think about the right methods how to model these to make predictions, okay? And we have many challenges there. There are many differences when you go from one hospital to the next, out of distribution differences, and we think about foundation models, how to adapt that, and so on. And then we test this in intensive care. We started implementing intensive care, so early warning systems in the intensive care unit, where we have a clinical study where these algorithms run in real time, make predictions, and give notifications to the physicians. I'm not going to talk about this further, but if you are interested in that, talk to me later. So I'm going to talk about cancer multiomics today. So let me start with this slide. So this is actually a slide which I've done in 2013. And it gave a bit of a vision of what we thought we should do in cancer. And we have all this interesting multimodal data, health records, pathology images, genomics data, drug information, and so on, right? And each data type is interesting in itself, in a way, but we need to distill certain pieces of information out of this in order to understand what's wrong or what's good about that patient, what's special about this patient, right? So these kind of pieces of information, they need to come together in order to develop something which we said is a latent representation of the health state of the patient, or maybe of the tumor if you want to treat the tumor. And then we can visualize this information, we can maybe predict some clinical phenotypes, and we can maybe predict some phenotypes over time. And maybe we could even find similar patients and make certain predictions of what happens with this patient or how to treat this patient. This was the idea, right? This was a nice grant, which was not funded. Anyhow, we continue doing that, and we still do that in some sense. This was part of the motivation why we involved ourselves in a study, and this is where Kyung really played an important role in co-leading this with me on the data science side. And this is the tumor profiler study. This is a study which was partially funded by industry, Roche, and of course, happening within academia, in collaboration between ETH Zurich, University of Zurich, and then two university hospitals, Basel and Zurich. And the idea was that we would like to test new technologies, profiling technologies at that point, where we take the tumor of a patient and then do very deep multimodal profiling of these tumors. And I mean, it's a really nice data set. And you get, of course, the electronic health records, the digital pathology images, you get targeted NGS. We didn't have whole genome for whatever reason. There's cell-free DNA, there's a single cell RNA, DNA, bulk RNA, proteomics, single cell proteomics, CyTOF, imaging mass cytometry, pharmacoscopy. This is a single cell drug in vitro test, and this is a multiplex drug response technology. So this is the data which you would get, okay, for almost every patient, not all patients. Sometimes we didn't have enough material. You would then try to get the information out of that, of these data sets, integrate this into what we called a molecular research report. This was like a little research project per patient, okay? So we try to understand what's good or bad about this patient. What's the response to a certain drug and so on. So a little report, actually not so little, a multi-page report came out of this data for each patient. This then went into a summary report, and this was then discussed in a pre-tumor board, which then informed, at least for some patients, the treatment decision. So this was one way. Actually, this whole process happened 240 times, and each of these loops happened within 4 weeks. So we could actually use this information for treating patients. It was a huge effort. I mean, there were essentially 100 people involved for generating this data. Each of these technologies had a whole group of people. There was a whole data flow which needed to be organized, which didn't exist, and so on, right? So this is, I think, where we really were pioneering doing this multi-omics technologies on these tumors. So the second level of analysis was then, and this is still, well, happening in some sense, but because not all is published, but we do the cohort analyses where we analyze these kind of patient cohorts together, right? So in order to see, is there something specific about these patients? Is there some new biology which we can detect? Okay, so this worked like this, right? So a team of scientists and oncologists, they all work together. We had these different technologies. Some of these technologies were high content image analysis techniques. So we got per patient, in some cases, 500 gigabytes of data. Then there were algorithms, sometimes AI involved, but at a low level, maybe image processing in order to detect cells and classify cells, how they behave, and so on. This was then summarized, and some derived data was obtained. Then this was selected by clinical scientists to form this molecular research report. Okay, and then at the end, so this was here mostly automatic. There was some manual parts in this, but this last part, this was mostly manual decisions. This was the discussion of the clinical scientist with the oncologist, among the oncologists, and so on. So as you can see, there's quite a few people involved here. And I mean, we did this because we were, let's say, well-funded in this case. There were, as I said, 50 people maybe involved in doing this, right? But you can also see this is probably not what we can afford in the future, right? So we probably need much better techniques to do this. And we need probably AI to make this much more efficient along the way. What came out at the end is a kind of a treatment recommendation table, or a table of possible treatments. In this case here, it was a ranked table together with whether this drug was approved for this patient, the different evidence levels for this treatment recommendations. And we also recorded the different in the different discussions in the tumor boards, we recorded what information was actually used in order to make a decision which of these treatments was used, okay? So we said, okay, in order to decide for this drug, we looked at this and this piece of information in order to support this decision. And this was very useful later, I show you. So in the end, we did about 116 melanoma cases, 80 high-grade ovarian carcinomas, and 40 AML patients. So there are marker papers, and which are, some of them are published, some are still submitted, and the data release actually, you can actually request the data now. So that's new. Okay. Good. Let's talk about these melanoma patients. So this is one part which is was published in Nature Medicine earlier this year, I think in May. We looked at 116 patients. We had 126 samples. We had 10 samples initially. It was a ramp-up cohort. We needed to understand this kind of data because we hadn't seen it. And then we had the 106 patients which went through this whole process. Among those, 13 were adjuvant treatment patients, 45 palliative patients, and 37 beyond standard of care patients. Why do we care about this distinction? The beyond standard of care patients are the ones where we are allowed to make decisions based on, let's say, this kind of research data. All the other patients, we have to follow standard protocols, the standard of care. So we could essentially not influence these decisions with the additional data, but we could influence the decisions in these cases because there were possibilities to do that. Okay, so what did we find? So first of all, maybe the data. So as I said, we have the sample processing, we have the biomarker results, and then we get to the molecular research report and so on, all within 4 weeks. usually it was quite stressful. We had to really time it well. The tumor had to be obtained, or the tissue had to be obtained on Monday to Wednesday, I think. So the results could be obtained within that week, and it was possible, but a little hard. In the end, we got out of the 106 patients, we got in total 103 total samples where we got information. We did not get a full, all samples, every technology available for every sample. Single-cell RNA, sometimes the cells were not viable enough and it didn't make sense to profile it. But in the end, we had a good multimodal data set where we had maybe a bit more than 40 patients where we had all the data, and then we had a good number of patients where we had a lot of the data, okay? And we had a certain priority here. We wanted to have imaging mass cytometry is something which we could always do. This is just based on FFPE. CyTOF doesn't need much material. We could essentially always do. Pharmacoscopy also doesn't need much data. It just needs maybe 100,000 cells, which can be distributed over the wells and then tested for different drugs. And then maybe prototyping, single-cell RNA, and so on, if we had enough material. But you can imagine there were a lot of discussions which technologies go first here, right? And this was the outcome of these discussions. So I said that we recorded for every single treatment decision, for every patient, what the recommendation was and also which information was used in doing this recommendation, right? So, and what you see here is essentially a graph. It shows one column is one patient, right? And one row is one piece of information. And it shows essentially which information pieces are used most often. And it turns out there's a lot. I mean, these are melanoma patients. Usually there's immunotherapy involved. There's a lot of immune-related information, but also there's a lot of spatial information, which is obtained. For instance, here the PD-L1, the T-cell infiltration, PD-1, TLS phenotypes, they are all related to digital pathology and IMC. And then there's a few other ones, of course, which are more genetics, but usually it's quite rare, so they are not used that often. So these immune phenotypes and the spatial phenotypes, they are used much more often than the individual somatic mutations for a tumor, because they don't happen that often. Okay, so one question you may ask is, do we need all these 10 technologies, right? So, because it's quite expensive. And you may ask, okay, so since we have this recorded, could we maybe compute which technologies would we actually need in order to make the same decisions? Okay, because we have recorded for this decision, we need this piece of information, and we would assume if we have this piece of information, we would come to the same decision. Of course, it may be fragile, but it's an assumption. But if we don't have this information, we probably would have come to a different decision, right? So, and what this plot here shows is every dot is one combination of technologies, a subset of technologies which are obtained. And then the question is asked, for how many patients would you have had the same information available which was used for decision making? Okay, so and what is interesting is, I guess, this curve here. So how many, maybe for the best combination of technologies, how many patients would you have had decided the same? So out of, in this case, 39, okay, these were a subset of the patients which were the beyond standard of care patients. These were particularly interesting. So you need essentially, if you have all technologies, maybe you need almost 20,000 Swiss francs, right? But you could do it with a subset of the technologies. So this would be NGS, pharmacoscopy, this is the single cell drug testing, and IMC imaging mass cytometry. So with this information, you would get 38 out of the 39 samples, you would do the same decisions using this model. Okay, there's a few assumptions in there which I'm simplifying, but you get the idea. So I think we can only do this kind of analysis if we have more information than we really need, right? So that's why I thought this was an interesting plot to look at. The other question is, how often do we actually benefit from this information which we have in the tumor board or in this extra multimodal data set? So what we did is the experimental setup was like this, right? So we recorded for the tumor board discussion 3 decisions. What is the decision if it is just pathology based? So based on the pathology report. What is the decision? This is level 1, okay? What is the decision if they could use the pathology information as well as, let's say, somatic mutations, FMI, that's in this case it was an FMI Foundation Medicine report. This was level 2. And level 3 was then using all the information which we had as a specific piece of information in Tumor Profiler. And then we could compare what was this decision based on these 3 levels compared to what was finally decided as the best way going forward, okay? And you can see is that if you have all this information, it agrees with essentially 75% of the final recommendations. Now you could say, yes, maybe the oncologist just liked this project and really liked this information and always went this way. That's a valid criticism. So in the end, we don't know. Maybe it's just the preference of the oncologist, but it seemed like there's additional information there. So the other question is, did it actually benefit patients? And to decide that conclusively, we really need to do a randomized controlled study. We didn't have that. It was a study where we essentially defined inclusion criteria. We took patients more or less randomly, and what we could do, or what we did in the end, is we went to the same period and found a matching cohort of patients, of real-world patients, and could compare the outcomes. And if you look at this study, you see a little bit of a signal if you look at all patients, all palliative patients. But the difference is not that big. So here you see the Kaplan-Meier curve, right? So this is for, 59 patients. And you see the 2 pro cohort fared a bit better, okay? But if you drill down a bit further, and you only look at the patients which are heavily treated, those which had already 3 treatments, then it looked much better. Okay, so because these were the complicated patients where essentially the oncologist didn't quite know what to do anymore, and then it makes sense to get this additional information, use maybe all that extra information about which drug may work on this patient and so on. So we find significant differences, but it's a quite heterogeneous cohort. So it's significant for the heavy pre-treated patients. But we really need a randomized controlled trial, and that is running now for ovarian cancer in Tumor Profiler. Okay, good. So now, so far there was not that much AI in there. So we helped this project, and it was really important that we were involved because we could help designing maybe the data collection much better than otherwise would have happened, right? So, and our aim was to bring in machine learning into this process much deeper than it is there before. So, you know, this process involved the sample analysis, okay, so that just needs to be done, right? So the bioinformatics analyses, these were mostly automated. Then the biomolecular interpretations and further analyses, this was mostly manual, and the oncological decisions, this was mostly manual. So what can we do? So I think we have 2 steps here, and they come at a different level of difficulty. The first step is that we get more relevant information out of the molecular or out of our data which we can have, right? So maybe we can predict some of the more expensive data out of the cheaper data. So that's why we started working on H&E. H&E has a lot of information, and maybe can we use that in order to predict other data which is expensive. So maybe find connections between data sets and get as much as possible information out of these data sets. So distill better information out of it. And the second one is, and that's I think much harder, and I think here is more the AI agents, which can really help, but it's really hard. So we would like to imitate what the oncologist suggests, maybe learn to rank these treatments based on that information which we distill from the data. So I will mostly talk about step one. I will talk about a project where we aim to achieve step two, right? But that's in the future. Okay, so let's get started. So I told you we have this data. Some of this data is spatial, and some of this data is single cell. And for us as data scientists, this was really interesting because we could get access to a very nice data set with maybe 100 patients. It's not that big, but you know, when it's single cell, it's quite nice. So, and what we did is over the years, and Kyung contributed to these things a lot, we developed a technique, for instance, single cell integration via deep learning and matching. So we had single cell RNA data, we had single cell proteomics data, and we found a deep learning technique which were embedding these in the same latent space, and we could essentially, for a single cell proteomics cell, we could find the corresponding RNA-seq cell. So we could match them, even though it was not jointly measured, right? So, and that was an interesting technique. We published in 2020, was one of the first ones. Then that's one which I'm going to talk about later. It's a multi-V stain, or we later called it histoplexer, where we predict protein markers from H&E. Then deep spot, that's predicting spatial RNA from H&E. I will talk about these 2 points. Cell OT, it's single cell perturbation responses in melanoma. I would like just to mention briefly what we do here. So we had these single cell measurements before and after treatment, and we developed methods which were essentially mapping cells before and after treatment, because you usually were not observing a single cell before and after treatment, but only a few cells before, a few cells after, and you didn't have the correspondence. And this was an algorithm to reconstruct that correspondence, and that made it really easier to analyze the single cell data. There's a lot of details I could talk about, but I don't have time. And then the last one is path of a probabilistic pathway multifactor analysis. I think this is another one Kyung was driving forward. Also very interesting. So I will talk about a multi-V stain and deep spot. So time, okay. So second part, multi-V stain. Well, histoplexer, what we also called it later. The idea, maybe in both of these projects, which I'm going to talk about now, is that we have a technology which is cheap and always available, like H&E. And we would like to use this technology in order to predict things which are expensive. And IMC is imaging mass cytometry. It's a nice technique because we can measure interesting markers, protein markers. We can measure phosphorylation and everything, right? So it's not covering large areas of the tumor. It's only tiny spots, tiny areas, and we can only measure maybe 40 proteins. But still, it's a very interesting technique. And the idea would be that we learn a relation between the H&E image and the IMC images, such that we can later predict these IMC images on the whole slide level. Okay, so we can also apply this to new H&E images. So essentially, can we skip the whole IMC measurement? So, but even for the same patient, it's very useful because we only have a measurement for a small region, right? And here we can actually generalize this to the whole slide. Okay, so yeah, before I go, so this is, before I forget, this is the work of Sonali Andani. She's finishing soon, I think later this year, her PhD, and Joanna and Bochi and a few others who contributed a lot to this, and this is collaboration with Viktor Kölzer, the pathologist who's now in Basel. And Bernd Bodenmüller and others contributed to the data collection and interpretation. Okay, so the histoplexer data set looks like this. So we have a tumor block. We have a normal H&E, the diagnostic H&E, which is obtained. Then we have an IHC for some tumor markers, I believe. And then we have IMC for a small set of regions, okay? And in total, we had 78 samples, which we looked at, where we had IMC. We had 336 aligned regions of interest. These were just 1 by 1 micrometer, pretty small. And we had essentially the pairing between this region and the H&E region. So we had essentially the multidimensional image plus the H&E image. But they are not super well aligned because they are 2 cuts away, right? So this alignment here is not perfect. So we try to align it as good as possible, but there are slight differences. And this also motivated the approach which we have chosen in the end. So we multiplex 11 protein markers, melan A, of course, S100, SOX10, and a few immune markers, which were very interesting for immune phenotyping, and then a few antigen presentation genes. Okay, so the workflow was like this, right? So we had the whole slide image, the H&E. Then we had the tissue image in IMC space, but only these little spots here, right? So these small regions. And typically we had these regions chosen in the tumor and another one at the boundary of the tumor to get to interesting regions. So we had these IMC patches. And then we had an algorithm, which I describe in the next slide, which took these H&E patches and these IMC patches. We trained an algorithm, and then we can predict IMC patches from that. And then once we have that predictor, we could use it to generalize to the whole slide. And we could do immune phenotyping, cell typing, survival analyses, and these kind of things, okay, which I describe in a second. Okay, here's the algorithm, how it works. So it would have been much easier if we had H&E and IMC from exactly the same cells, but we didn't, right? So because it was the next slide. So what we could do is we used what we call a generative adversarial model, a GANs or network, which takes an H&E image and it tries to predict the IMC image, but we cannot exactly say which IMC image. We just would like to have it indistinguishable from IMC images as they look like, right? So, and essentially what you do is you put in this tile into the predictor, predict the corresponding IMC image, and then have a discriminator which says, oh, this looks like a real one, or this looks like a bad one, right? Or this looks like an unreal one, so a fake one. In this way, these networks learn how to generate realistic IMC images. So, okay, so this is the GAN approach. You needed a few more things because we didn't have good alignments. We needed to generate losses or differences. We needed to measure differences at different scales, right? So, and here we used a pyramid loss with some Gaussian filtering, where we measured the correspondence of the predictions with the real images at different scales. So at high resolution, medium resolution, and lower resolution, and so on. So because, yeah, if they are not exactly aligning, the pixel is not aligning, so then the loss is just high, and you can't do it, right? So this was one other technique, and the last technique, it was a contrastive loss, where we try to use the relationship of patches in the same image and try to discriminate them against images or patches in other images. So this is a typical machine learning approach. So does it work? I think it does. So this is the input image, an H&E. This is the ground truth. This is what we measured. These are 6 of the 11 channels which we had. Melan A, CD3, CD8, and so on, right? And these are the predictions. So this is the test set, right? So it didn't see these before. So, and it looks pretty good. So you could measure that in different measures. We distinguished between multiplex prediction and singleplex prediction. So you could do essentially the same method in predicting a single channel. and you can do this prediction based on multi-channels. And the question is, okay, a single channel maybe just good enough, right? So it turns out if you learn it jointly, so you do multiplex prediction, then histoplexer, the same technique, right? So it's the same model, everything. Multiplex works significantly better than the singleplex model. And we compared it against pigs-to-pigs, pyramid pigs-to-pigs, and so on. So it was significantly better in every measure which we looked at. So that was promising. We also showed that when we do multiplex prediction, then the correlation structure between markers locally is better preserved. So we looked at every marker combination, okay? So for SOX10 and HLA-DR, when you look at the spatial correlation of these two markers, then they are usually anti-correlated, okay? And that is what you can measure in the real images. And what you would expect is that in the predictions, the correlations should be more or less the same, right? So, then you can measure how big is the difference between the correlations for these pairs between this multiplex and the singleplex predictions. And you see the multiplex predictions, the mean squared error between the correlations is much smaller than for the singleplex. So I think it's much more faithful in producing these correlations. Okay, good. So now, how can we use that? So we could now take the H&E image, we do the protein multiplex prediction, then we have cells annotated, and we could simply assign to each of these cells the corresponding protein expression level. Then we had, in IMC, we had cell type classifiers, which was using the cell expression, and we simply applied that to the predictions. So no fine tuning whatsoever. Just the rules which were developed before were just applied. And we could then distinguish tumor cells, CD8 cells, and so on, right? And that's, of course, very useful. We could say, what is an immune hot tumor? This is a tumor where T cells, CD8 T cells are infiltrating the tumor. And this is one of these cases here. You have tumor cells, you have a lot of CD8 T cells inside the tumor. That's a hot tumor, and this here is a cold tumor. And you could quantify that and then really see that this makes sense. And we compared that against previous annotations of these tumors by pathologists, and that was very well agreeing. Yeah, so maybe I skip that. Good. So we also applied that to a different cohort. You could say, so maybe the tumor profiler cohort is very homogeneous and maybe it cannot be applied to patients outside of tumor profiler. And it's a valid concern. I mean, we deal a lot with covariate shift, domain out of distribution generalization. And we were surprised that it works quite well. Not perfect, but quite well. So we used the TCGA data. These are the FFPE TCGA data. We generated whole slide protein multiplex images from H&E. And essentially used two ways. We tried to build a model which is only using H&E in order to predict something. Okay, and we used another model which used H&E plus the predicted multiplex images in order to predict, let's say, survival or other properties. And so essentially, we worked on two downstream tasks. One is survival prediction, the other one is immune subtyping, and one is single modal, is only using H&E, and the other one is multimodal, which is using the predicted multiplexes. So, and we think that these predicted multiplexes, they have much more information, and it's much easier to extract something informative out of it. So, and that's what I would like to show you. So if you would take the immune subtyping, then you could go from low, intermediate, high. Let's say you obtain a label based on RNA-seq, which is not given to the algorithm, right? So that's our ground truth label. And then we measure how good can our model predict the RNA-seq-based immune subtyping phenotype. And we see that with the multimodal model, if we use the predicted IMC images, we are significantly better in terms of accuracy of predicting these labels. So this is the unimodal model, this is the multimodal model, and here you get to 82% or so, and with the unimodal it's just 65%. Similarly, if you would like to predict survival just based on the H&E, you're also significantly, well, a bit worse. The difference is not as big. So, but you see, you also get better when you use the information from the IMC predictions. I mean, it's maybe surprising because the H&E has, I mean, we use the same input, right? But we just extract more information out of this image, and then downstream it's more available for making predictions. And the same, we think, would be the case if we give this information to a physician where he or she can see this much more clearly, right? It is in the H&E, but maybe an expert pathologist would see it, but maybe the oncologist not anymore. Okay, so summarizing this part, getting closer to the end of time, not to the end of the talk. So, yeah, I think histoplexer uses morphological information from clinically established pathology images. It shows that multiplex prediction approaches are consistently outperforming other singleplex alternatives. And we think that prediction on the whole slide level is really informative for spatial analysis of the tumor immune cell interactions. Okay, so this was published maybe a month ago or so. You find the reference here. Good, code is here, data set is here, and so on. Good, so let me, considering the time, maybe I shorten this part a bit. We started late, oh, that's good, good news. So then let's talk about spatial transcriptomics. Just 1 second. So you have probably seen this this example. Would be nice to have this fruit shake now, right? So, also this one is really nice, and probably you've seen this one too, right? So, I mean, this shows bulk RNA sequencing, single cell RNA sequencing, spatial transcriptomics. So this is about spatial transcriptomics, which is great because you see a lot of spatial information, really good. However, spatial information is really small. It's just 6.5 times 6.5 millimeters. It's tiny, it's complex, and it's expensive, right? So it costs, I don't know, for a single sample, $5,000 or so. So same idea. If you would like to take the H&E image, have some kind of regression model, and predict the expensive spatial transcriptomics data. So very similar. Just different type of data, different approaches, because here we have better alignment of the spots to the image regions. So this work, again, involved some of the same people, but it was really driven by Karlin Nonchev, a great student. So, and at ETH. So here's what he designed. So we took these H&E images, we put them into tiles, and for each tile, we used a foundation model for pathology. So these are pre-trained models which take a piece of image and produce a latent representation of that image, which then can be used further. Usually these foundation models, they are trained on millions of images, or at least hundreds of thousands of images, and they contain a lot of, let's say, knowledge of what these cells mean, right? So, and what this approach is doing, it is using that as a first layer to process information, because our spatial transcriptomics data sets are never large enough to learn that. So we use these models. We first apply it to the spot itself, okay? So to the region where the spot is. Then we split that spot into 4 regions, apply that model again, and then we use the tiles which are around. So essentially this creates a bit of a spatial neighborhood, a bit higher resolution, a bit lower resolution, and get different representations from the same model, okay? And then we use what we call a deep set neural network, where you have these representations, you combine them into one representation, and then you use an MLP essentially to predict the spatial transcriptomics, the gene expression here. Okay, so that's essentially it. It works. Let me show you how good it works, but I don't want to spend too much time. This is the H&E image. We have also an annotation of that H&E image by pathologists. They say, okay, here's tumor, most of it is tumor. There's a bit of stroma, there's some lymphoids, and there's some blood necrosis. And then we can measure essentially how good is the correlation, the spatial correlation for each of these genes which we predict. And this is here the SOX10 gene, which is relevant. And this is a method which is called BLEEP, has a Pearson correlation of 57%, and a deep spot has 70%. So, and you can see, the structure matches very well with the known annotations here. So we can also measure this in more detail. So now, how good does it work over many genes? And here we look at the most variable genes. So these are not the most predictable genes. These are just the most variable genes in the data set, okay? And then we try to predict the gene expression spatially and measure the correlation. So mind you, the correlation is not super high, right? So it is 20%, in some data sets it's 40%. If you would sort by the most predictable genes, it would be significantly higher. So then there are some genes which you can predict with 60, 70%. But yeah, I think it makes sense to look at the most highly variable genes, and then the top genes you can predict relatively well. And when you get to the tail of the gene expression, it gets lower. And that's not surprising. How much can we actually see based on the morphology of a gene, right? So how much can we see based on maybe the neighborhood of that cell? It's clear that we cannot see everything, but we can see something, right? And this is what it shows, right? And it shows the technique which we have chosen works quite well. So it works significantly better than other approaches which are out there by quite a margin. So this is for a colon cancer, just trained on eight images. We also did this with a tumor profiler, melanoma, kidney cancers, and lung cancers. So, and it was essentially the same picture in all cases. Yeah, we don't have many samples here, right? But we have many spots, right? So, we can still generalize, so it's okay. But these measurements are always trained on n-1 patients and tested on the last patient, or the held out patient. So here's another way to measure. We looked into TCGA. For TCGA, we have usually two images, H&E images. One is obtained from FFPE, and the other one is obtained from fresh frozen tumor. And it's usually pretty bad quality in terms of histology image, because these are frozen cells. They're kind of half, they're dead, but they're destroyed by freezing. Typically people tell you, well, these are not good pathology images, so you can't use them. But still, what we see, okay, so maybe before I explain this figure, so we measured then how good can we predict gene expression based on that image, and we can compare that with bulk RNA gene expression, which was obtained in TCGA, okay? So we could compute essentially the correlation between the gene expressions predicted versus bulk RNA. Predicted pooled, right? So because we have prediction for many cells. And then this is the correlation again over the most variable genes. So there's one interesting bit, I think, first the method works good, so you see that, but what I found was really interesting is that the fresh frozen images were much better aligning with the gene expression. When we predict based on the fresh frozen tissues, then we predict gene expression much better, or it's much better aligned with the gene expression which was measured. And anybody an idea why that is? Yes?
The asymmetry, right? The distribution of omics would just be formed by the
Yes, indeed. So essentially, the bulk RNA was exactly obtained from this block, right? So therefore, this is the right tissue piece, right? If you have heterogeneity in the tumor, then the FFPE and the fresh frozen they would come from different regions, and then the match is not as good, and that's what you see, and that's quite consistent. Also, when you look into the other tumor types, there's usually quite a gap between the fresh frozen and the FFPE. So it doesn't mean the FFPE is bad, right? So it's just a different region of the tumor, and that's why it's not matching. And why does it still work? Usually you would say, okay, the FFPE, the fresh frozen images, they are not good quality. But the foundation models, they have seen so many images that they can really abstract from this, so they're not distracted too much by the bad quality of the image. That's our interpretation. Okay, so let me show you maybe some additional bits here. So you could, what else you could do? You could maybe try to predict survival. There's the C index. We use the Cox proportional hazards model with the elastic net penalty to use bulk RNA data to predict survival in skin melanoma and in Kirk, that's kidney, I think. And what you can see is if you use fresh frozen, it works pretty well, in particular when we use our predictions. If you use FFPE, it works a bit less well. Here for this one, I don't know. Here the interpretation is different. Anyhow, so you can see it works usually. Deep spot has the best performance. Sometimes there's a difference in fresh frozen versus FFPE. Okay, maybe one thing I would like to mention. So I'm somewhat proud of the student, so that's why I mentioned it. So he participated in a broad challenge for predicting spatial transcriptomics based for an autoimmune disease. And there were thousands of participants, and this deep spot approach made the first rank. So this was really nice. So it was really showing that it's not just our evaluations, but it is also performing well when it's evaluated by others. So now, what can we do with this? And Karin has been quite, let's say, creative in thinking what else can we do. And one thing is we could take all the H&E images out there and just predict the gene expression, right? So, and then this would allow maybe some additional analyses which otherwise we couldn't do based on this. So this would give us spatial information for thousands of images based on these models. And we have done this for some of the tumors where we had spatial transcriptomics data. We could learn models, and then we could apply it to the corresponding cancer type where we had these images. So, and these images, they are available on Hugging Face, and you can download and analyze. And you can do, I mean, many different types of analyses. You can cluster the regions, you can detect markers in different regions, you can do pathway analyses. With these artificially generated H&E expression maps, right? So, and we show in the paper that you can do certain analyses on these predicted expression patterns, and they lead to very similar conclusions as you would have had done if you had the spatial transcriptomics data. So this was, I thought, surprising. So maybe one final bit here. We started collaborating with the people from the Bock lab in Vienna. They have a tool which is called Cell Whisperer. It's essentially a tool which combines gene expression data with a large language model. So, and it's a single cell data set. Let's say if you would put in a single cell data set, then you can ask the language model, show me all the immune cells. It would figure out what immune cells are and highlight maybe all these cells. Now you can do the same and apply that to an H&E image, right? Show me all the immune cells on the H&E image, and it would highlight them, right? So, and that is, we did as a proof of concept and did a prototype, and that worked quite well. So if you go to that link here, you find a little video and a little demonstration where you can do this. You upload your H&E, it runs through the deep spot model, is then interpreted by the large language model, and it helps physicians or researchers to understand the H&E image, which otherwise, I mean, I typically don't understand them, right? So this would really help me. Okay, so getting to the end of that part. So in summary, DeepSpot is a new approach to effectively predict a spatial gene expression from H&E images. It improves the accuracy and granularity of gene expression. And I think we have generated the largest digital multimodal spatial transcriptomics data set, a virtual one, right? It's not real one, but which has over 56 million spots from melanoma, kidney, lung cancer patients. So with that, let me come to some conclusions, but I have a few conclusion slides. Okay, so in general, tumor profiler and multimodal data, they show a great promise, and we think they can make a difference in tumor boards and may improve patient outcomes. So at least we have some evidence that this looks good. But we do need randomized trials for ovarian cancer. This has started, and we need it for the other cancer types too. So the large single-cell and spatial multiomics data sets, which I've described, they really bring many opportunities. And I have to say that I benefited a lot from this project because we had access early to these data sets and we could develop these techniques. So there's many biologic insights which are reported in the marker paper, which I have not described here, but soon these will come out too. Histoplexer predicts a spatial protein expression, DeepSpot spatial transcriptome. What I have not shown is methods for predicting single-cell drug responses. Now, you could imagine now that you have your H&E image and you can predict gene expression, and from the gene expression you can predict which drugs will work. And then we get kind of a map which tumor part or which part of the tumor would be attackable with a drug, right? So, I mean, all has to be validated, but as an idea, I find it fascinating that we could do this, right? So that we maybe understand the subclones of the tumor and how different parts of the tumor would actually be treatable with different drugs, and maybe which drug combination we have to take in the end. So this is a nice data set. We worked hard to analyze it in the first place, but also to make it releasable. And I think you can, if you go to this web page, you can request the data now. And it will be available under controlled access. So let me finish with some thoughts about AI-supported molecular tumor boards. So molecular tumor boards involve high-stakes decisions, and usually there's very little time when these decisions are made. In these molecular tumor boards, which we discussed, they only had 5 to 10 minutes per patient. And a lot of data needed to be prepared by a set of other people in order to make the decisions, right? So, and in Tumor Profiler, we had this large team of experts that helped with the interpretation, but this is infeasible, essentially, if this is not well prepared, and you usually cannot do this much effort. And I think here it's really important to push the limits from what we have done with manual interpretation into automatic processing and automatic interpretation of these quite complex data sets. Also to see that maybe a certain signal I see in one data modality, I also see it in the other modality, and point this out to the physician that there's some kind of validation. So we think that AI tools can extract more information from these clinical routine data. It could also extract more if you had other types of new data types, sure. We think that AI agents can maybe find related policies which are in the hospital, related publications, or maybe other patients which are like that patient more easily, and could summarize this molecular data in an easily digestible form to the oncologist. I mean, that's a vision. There's a lot of work before this can happen, right? Because a lot of bad things could happen here too. But I think there's an opportunity here which we have to develop. So I think in the near-term future, I think the oncologists will make the decisions, but they will get more and more AI support from this data. So the research really has to show how to make these AI tools accurate. the AI agents safe and trustworthy, and how to make them even better than maybe the average oncologist. So that would be the goal. Maybe very briefly, so we have started more into the future thinking. We have started a project which was just announced a few weeks back. It's called the Swiss National AI Platform Oncology, and it's essentially a project where we try to implement some of these things, right? So that we bring some of these tools into the tumor board to the attention of the oncologist such that they can use this information. And it has different pieces. It needs language models to understand to understand the clinical practice documents and in the literature. It needs methods like the ones which I've presented for the clinical routine data, and it needs AI methods for clinical discovery. So, and these kind of things need to come together and be presented in the AI tumor board, which present these predictions, summarize information to the oncologist, and so on. So there's a lot of work to be done. It goes from infrastructure building such that the data can flow, a lot of complexity. Some of the algorithms which I described today would be usable here, but we need other and better algorithms to do that. Maybe I skip this here in the interest of time. So maybe just some meta conclusions. I mean, for me, I've always very closely interacted with physicians or life scientists, and this really made kind of the type of work which we do, I mean, maybe fruitful or also enjoyable, interesting, because we could deeply understand what the problem is about, understand what needs to be done here, right? So, and which prediction would actually make a difference. So, and I think if you're a computational scientist, this is really something which you ought to do, I think. So you need to understand what is used for. Often there is a good solution with some existing methodology, and sometimes it's the right thing just to take that and help maybe the life scientist in the first iteration. But then I think it's important that one goes back, maybe finds a better method, finds an interesting methods question that maybe can lead to a better solution, maybe lead to a new approach, a new technique, right? And for me, this cycle is very important. You need to understand what is relevant, maybe help out in the first iteration, but really understand how can this be done better. And this, for instance, with the cell OT, this drug response modeling, this was the case. We first had some techniques to analyze this data, and then only in the second iteration we had a much better technique to do that. Okay, and of course, yeah, don't stop when you have a nice method, but maybe also demonstrate a clinical impact. If you develop deep learning methods compared with a simple model, like a linear model, many people don't do that, and it's really important. The biomedical foundation models I've shown you today are very promising, but a lot of work still needs to be done here. And yeah, maybe as a general remark, healthcare and life science is full of out-of-distribution problems, and we need new methods in this area to deal with that. So with that, I think I'm sufficiently over time. I would like to thank my team. I'd like to thank you for your attention. Thank you.